Lori Sussel, who came to us from the University of Colorado, Barbara Davis Center, but who actually was a graduate student at Columbia before she went abroad, so to speak, and then came back.
Anything not in New York.
Exactly.
Lori is a professor of genetics at Columbia, but works in the Diabetes Center full-time. So her lab and her research activity are conducted in the Diabetes Center. And she is really, I think, one of the reigning experts on what would be described in a general sense as the developmental biology of the beta cell. Again, as you'll hear from Lori, if you ask her, the beta cell originates as a cell that doesn't produce insulin and gradually matures over a series of steps to become a competent insulin-producing cell. And there are 4 or 5 other cell types in the islet that orchestrate or play a role in some of the responses, at least systemically, to the metabolic changes that require insulin. And Lori really is an expert in the development of these various cell types and how they play on each other in terms of the biology of the beta cell. And in fact, Lori discovered what people thought was sort of like we knew all the planets. They thought they knew all the cells that are in the islet, and Lori came along and discovered another one. This is the ghrelin-producing cell, which is a very interesting cell in its own right, and maybe she'll tell you something about it. But for virtually all of the people who are on this panel, Lori is a go-to source for this developmental biology. That is, we need to understand if we're going to try to make beta cells from stem cells, make beta cells that can be studied in immunobiology models, make beta cells that come from the gut, make beta cells that interact with macrophages. We need Lori to help in terms of the informing the kinds of experiments, the kinds of processes that are involved in these various really quite complicated areas of biology. So Lori is a tremendous addition to the Diabetes Center now, what, 6, 7, how many?
6 years.
6 years, and it's really been an enormous addition to the center. So Lori, you can speak for yourself.
Well, actually, Rudy just said it all. I'm basically, beta cells is what we do. We use mouse as a model system, but we're very interested in how during fetal development, how beta cells are made. It's something, the regulatory networks we don't understand, and if we want to do work like Dieter's doing to make a stem cell into a beta cell, we really don't have all the tools available to us just because we don't have the knowledge. So what we're using is the mouse, which is very conserved in the regulatory networks with the human, to understand how during fetal development can you make a beta cell. And we think this is a really great model then to translate that into taking stem cells and differentiating them into beta cells. We've also now moved on because the stem cell biologists have really done a good job of starting to make insulin-producing beta cells, but they're not doing it very well, like as far as very efficiently, and also these cells aren't functioning as well as they could be. So now we're starting to look more in the adult and saying, how do you make a better beta cell? How do we make a beta cell that can sense insulin or sense glucose really well and produce insulin in response to that? And we're learning mostly how not to make a good beta cell, but we're hoping that if we start with, not making a good beta cell and then we can fix it. And so that's our whole goal, is to say, okay, how can we make a better beta cell? And so the research is basically focused on beta cells, how you get them in the first place and then how you can make them work. And then also, how do you keep them working? I think this is another major problem that some of Mimo's work has brought up, is that once you have a beta cell in an adult, how do you keep it making a good beta cell? And most recently, actually, for type 1 diabetes, we've been focusing more also on why is the beta cell a target? I think we focus a lot on the immune system, and we forget that the beta cell is uniquely targeted by the immune system. And so since we're now experts at beta cells, we decided it was a good time to start looking at what is different about these cells that make them targets.